The inside of the cell contains the cytoplasm, but within that cytoplasm, we also have an internal skeleton, and that internal skeleton is known as the cytoskeleton. This internal skeleton is going to be anchoring different organelles or different cellular compartments so that those are not just randomly floating around inside of the cell. They actually have a set position. And then it's also going to be important for cytoplasmic streaming and organelle movement. Cytoplasmic streaming is one way that we refer to the movement of things within the cell, so from one side of the cell to the other. So it is kind of a movement of the cytoplasm since the organelles are part of the cytoplasm. Now, as far as what the cytoskeleton is actually made of, is it's made up of a variety of different fibers, and these fibers have different proteins that compose them. Some of them are made of tubulin, and those we call microtubules. We have some that is made of actin, and we call those microfilaments. And then some of them are made up of a variety of different proteins, and we call them intermediate filaments. There are slightly different roles to the different types of fibers, depending on what they're made of. But overall, they are responsible for providing support inside the cell and then also anchoring and movement. We also have ways that cells can move, and these are what we call cell extensions. So by extensions, they're actually projecting from the cell. They fall into two main categories, although we will have a third type as well. The cilia are like little tiny hairs that we find on the surface of the cell. They're going to be modal, so they will be actually moving like this. They beat rhythmically, and they are like hair-like structures. You will actually have tons of these on the surface of a cell if they do happen to be present. This is not something that you're necessarily going to see in all cells or on all cells. The flagella are different in that these are single long tail projections. Now I say single, there may be a few of them there, but these are going to provide a wave-like movement, and they provide some really fast speeds and directional movement as well. So if we have a cell that is trying to respond or get close to some type of chemical signal, they can really help that cell to move really rapidly in that particular direction. One third or final category for cell extensions is what we call microvilli. And these are tiny finger-like extensions that you can see in this picture here. And these, if I were just to draw on here, they're going to create this type of an arrangement with the membrane on the surface of the cell. So the purpose of that is that it greatly increases the surface area of the cell, which is going to help with absorption. So in that way, it will be able to absorb a lot more materials much more rapidly than a cell that just has a nice smooth surface area to it. We have a lot of different types of cells inside of our bodies. There's actually over 200 types of cells inside of our bodies. And depending on their specific function, they might have a different shape to them. But overall, they are going to have the same general features, such as the nucleus and the different organelles that we've talked about. If we go through and show some pictures of some of the different cell types or some of the different functions that they play inside of our bodies, some cells are responsible for connecting body parts together. So they will actually kind of tie different body parts together. We have some that are responsible for covering and lining body organs, and you can see that those have a different shape to them. And we have some cells that are responsible for moving organs and moving body parts, and cells that are involved in our skeletal system are going to play some of those roles. Some are responsible for storage. So we have fat cells, for example, that do store fat, which is a form of energy storage. We have some cells that are responsible for fighting disease. So they fight different things that might attack our bodies or some just internal problems that we have. We have some cells that are responsible for gathering information, so pulling in information and responding in some way to control body functions. And that really is our nervous system that's doing that. And then lastly, we have some cells that are responsible for reproduction, which is one of those very important functions that living organisms need to perform. And the example that we see here is a sperm cell. And that one has to be modal because it has to get to that egg. And notice, because that is the role of that one, it actually has a long flagella on it to help it with that movement activity. So what all can cells do? Okay, so cells, we said they have basic functions that they have to be able to do so that they're considered alive, but they really have a long list of things that they're capable of. Cells are able to metabolize. This means that they perform different reactions. They are able to digest food, and we will have compartments within the cell that are responsible for that digestion. They're also going to need to get rid of waste, so they will have a waste disposal system. They reproduce, they grow, they move, and we've looked at features of the cells that help them to move. And then ultimately, they do respond to stimuli so that they're able to respond to their environment in whatever way is necessary. So in order to do this, it is going to involve a communication with what's on the outside, and then also communication inside the cell. Our cells are ultimately surrounded by fluid on both the outside and the inside, so water-based solutions. So important terms to know if we're talking about these fluids is that, first of all, it's a solution. A solution is a homogeneous mixture of two or more components, and that solution is made up of a solvent and a solute. So the solvent is going to be the substance present in the largest amount. So in other words, this is the liquid that's doing the dissolving, and then the solute is the substance that's present in the least amount, or in other words, this is the thing that is being dissolved. So we have a solution that is made of a solvent that contains solutes on the inside. And if we just compare this to, say, sugar water, if we have sugar water, the water is the solvent and the sugar is the solute because it's being dissolved inside of that water. So our cells have solutions on the outside that have water as the solvent and lots of solutes inside of them. And then on the inside, it's the same thing. We have solutions where water is the solvent and you have lots of solutes inside of that. If we're talking about the solution on the outside of the cell, the exterior of the cell, we refer to that as interstitial fluid. And then if we're talking about fluids and solutions on the inside of the cell, we call that intracellular fluid. Now the membranes, that's the outside of the cell, that plasma membrane. It is really separating that interstitial fluid on the outside from the intracellular fluid on the inside. And the membrane will ultimately be responsible for regulating the passage of molecules from one fluid to the other fluid. So regulating things on the outside, passing to the inside, and vice versa. So if we talk about more with regards to this membrane transportation and regulation function, membranes, one thing that they do is they maintain concentration gradients. Membranes are barriers, so they are going to keep the inside isolated from the outside, and because of that, they are able to concentrate chemicals on one side or the other. So they're keeping a certain concentration over here versus on the other side. In this picture right here, we have two different types of ions. We have some little orange ones that have a positive charge on them, and then you have some little green ones there that have a negative charge on them. And notice that what you have on the bottom versus what you have on the outside is a different concentration. So if we're specifically looking at the orange ones, we have more of them on the outside versus inside. And with the green ones, we have more of them on the inside compared to what we have on the outside. And this is typical when we're talking about membranes separating different compartments. We can have different concentrations of things on the two sides. Now, if we do have something that has a charge to it, and in this case, what we're looking at here with the orange and the green, there is a charge associated with that. Say that we also have an electrical gradient. And that electrical gradient is the charge difference, or what is also known as the voltage. Movement across the membrane is going to fall into two categories. So there's two types of transport. The first one is passive processes. Passive is just like it sounds. With passive, it's something that just kind of happens on its own. We don't really have to force it along. What describes the passive processes is, first of all, they do not require the cell to spend any energy. So we will not see any use of ATP to actually make these things happen. And then another thing that describes them is that the substances are moving from a high concentration to a low concentration. So if we sum that up, they're moving from high to low, or in other words, they are spreading. So you might have something that is ultimately really concentrated, packed together very tightly, and with the passive process, it will be able to move and spread out. There's different types of passive processes. Here we see a description of one of them. This is diffusion specifically, but notice how we start out with a lot of molecules on one side, and this is our membrane that's shown in the middle, but we have a lot of molecules on one side really crowded, and then over time they spread out and take up the space on the other side. So that's the passive processes. That's the moving of the molecules from where they're really at a high concentration. Over here, they're at a low concentration, and notice that they're moving in that direction. They go from where they are at a high concentration to a low, and eventually they become even on the two sides. When they do become even on the two sides, we call that then equilibrium. So examples of passive processes, first of all, is diffusion. Diffusion is the tendency of molecules, any type of molecule, to basically spread out and take up all the available space that's there. And this does happen with regard to cells, especially if we have molecules that are capable of just passing right through that membrane. Not all molecules are going to be able to readily cross the membrane, but certainly some of them can. Examples of ones that can do that would be something like oxygen. Our cells are in a constant need of oxygen, and oxygen will just pass right through. As the cells use it up on the inside, the concentration gets lower and more oxygen will flow in. So that's one example of a passive process. Again, notice that we're not saying that we have to really expend energy to get the oxygen to move into the cell. So this is simple diffusion. The substances have to be able to get through the membrane if they're going to go in by simple diffusion. Some molecules can't get through the membrane. If they're big, if they have a charge to them, if they're hydrophilic, they just can't get through. So with those, we have what's called facilitated diffusion. And facilitated diffusion, we are facilitating or helping them to get across. And the way that they're going to be helped is with the use of proteins. So here we can see we've got proteins shown in purple. These proteins will actually be stuck in the membrane, so they span from one side all the way to the other, so they really reach across. And these proteins work in a couple different ways. They can provide a kind of tunnel through the membrane, or a channel that runs from one side to the other, so they can just open up and now the molecule can flow through. Or in some cases, they actually grab hold of it on one side and pull it across to the other. So that's facilitated diffusion. Again, no energy is being involved here, and molecules are still spreading out or going from where they're really concentrated towards where they're less concentrated at. So substances with this one are going to use a membrane protein to help them get across. A third type of passive process is osmosis. And with osmosis, we are specifically talking about the movement of water molecules. This will be diffusion of water. So by diffusion, they are spreading out, and the water molecules are going to move through special proteins, which we do call aquaporins. So this is an example of the aquaporin right here. It's going to help the water molecules to get across. And the water molecules, again, move from a high concentration to a low concentration. Filtration is a final one. Filtration will be when substances are actually forced across the membrane due to some type of pressure that's being applied. And so if an organ is actually squeezing, so contracting, or the cells are contracting and getting smaller, that would help to force something from one area to another. The second type of movement or transport across membranes is an active process. Active is going to require more effort on the part of the cell. In this case, the cell does spend energy. It has to have energy to invest. And typically that energy that it will be spending or investing in this process is going to be in the form of ATP. So that's one difference between the passive and the active. The other difference is the direction of movement. With active processes, it is always going to be from a low to a high concentration. So in this case, we are actually taking the molecules and making them more crowded. So we're packing them in more tightly. And this one is always going to need some help from membrane proteins. If we look at the picture right here, we have a protein, the purple, that is actually moving two different substances, which can be the case when we're talking about active processes. And so with this one, we're actually moving sodium from one side to the other. And then after we do that, we move the potassium in the other directions. The potassium is the green one, or the blue one here. Notice that we do see some ATP being used over here. That's the energy source to make this entire process run. So active processes, we have to use some energy. We are concentrating or compacting them, getting them much more closely together, so moving from a low concentration to a high concentration, and we always have to use a protein to make that happen. Active transport is also going to include what we call vesicular transport. This is what happens when we're talking about movement from the ER, the endoplasmic reticulum, to the Golgi apparatus, or from the Golgi apparatus to the plasma membrane. This requires basically packaging things into a vesicle and then fusing that vesicle with a membrane that happens to be somewhere else. This is going to help us to move a whole lot of things at one time. So this includes both endocytosis and exocytosis. It's really all about the prefixes here, endo and exo. Endocytosis, endo means inside. So this is basically drawing things in. And we can see in this picture here that it can be a little puckering in of the membrane. It has particles inside of it. It pinches off, and we end up with a vesicle on the inside. When that happens, we have drawn in a bunch of substances that were on the outside. Now they're suddenly on the inside. There are some different categories of endocytosis, and this picture is just showing some of the different categories. But overall, we're basically drawing things in by forming a vesicle. Exocytosis is just moving things in the opposite direction. Exo means exit, so in that case, things are actually leaving the cell. So we take something that is inside the cell, we package it into a vesicle, and then we fuse it with the exterior. And when that happens, the contents are now kind of dumped to the outside, or what we can technically call secreted.